Hello, everyone. Welcome to our Cooper Surgical webinar on the ER Complete test. We are going to wait just about a minute to let everyone join the webinar. Okay, welcome everyone to the Cooper Surgical webinar for today. Join us today. We'll be discussing the latest research into the endometrial microbiome. We'll talk about the impact of the endometrial microbiome on embryo implantation. We'll discuss the ERPeak test and the demonstrated improved outcomes that have been seen for patients with recurrent implantation failure. And we will discuss ER-Complete, which is a 3-in-1 test to assess when is the right time and the right environment for a patient to have an embryo transfer. So we'll start with a little housekeeping just to get started. First, please be aware that the webinar will be recorded so that it can be shared in the future. Second, everyone except for the presenter will be muted. However, you can still interact and ask questions using the Q&A box to the right of your screen. We really urge you to take this opportunity to get involved. We definitely want to hear your questions. And at the end of the presentation, your questions will be selected for discussion, and any unanswered questions that we don't have time for will be forwarded to the appropriate team members for response after the webinar. So let me go ahead and introduce our panel for today. First of all, I'm Jenna Miller. I'm a genetic counselor by training. I'm our clinical science specialist at Cooper Surgical. And then our panelists for today are Tony Gordon and Kristine McWilliams. Tony Gordon is, he has a PhD in molecular cytogenetics. He has over 20 years experience in molecular diagnostics. He worked at the Institute of Cancer Research in the 1990s and at various companies in the diagnostics field. He joined Bluenome in 2006, and in 2013, Tony joined Genesis Genetics, which you may remember as a leading global company for pre-implantation genetic testing. He was managing director for their 5 US laboratories and lab director for their 2 labs in the UK. And in April 2016, Genesis Genetics was acquired by Cooper Surgical, and that is where Dr. Gordon is today. He's currently our Senior Director of Clinical Strategy and Market Development. We're also lucky to be joined by Dr. Kristine McWilliams. Dr. McWilliams is the Medical Director for Genomics here at Cooper Surgical. She is instrumental in defining the path of genomics, of the genomics division. She works closely with cross-functional teams, including our genetic counseling team, which I'm particularly partial to. Kristine manages that team and guides the team's clinical decision-making. She really tries to ensure that clinicians and patients are equipped with genomics information that would positively inform their transfer decisions. She's been working in the field of PGT since 2013. She has particular expertise in PGT-M, She obtained her MD and PhD degrees from the University of Pennsylvania, and her PhD work is in the field of human genetics and epigenetics. And Kristine is going to be our first speaker today. She'll be discussing the endometrial testing revolution, as well as our ERPeak test. So I am going to turn it over to Kristine now. Take it away.
Okay, Jenna, thank you very much for that introduction. I'm really glad to join everyone today to talk about the topic of endometrial receptivity, the endometrial biome, and our new ER-Complete test. I will be focusing on telling you a little bit about where we stand right now on the topic of endometrial receptivity and our ability to test a patient's own endometrial receptivity via omics and specifically transcriptomics. Endometrial receptivity is something that's been studied for a number of years. However, until just a couple of years ago, it's been primarily a visual science to assess the quality of a patient's endometrial lining. And we're excited to move into this phase of omics testing for endometrial quality. And I'll be talking to you about that. So I'm excited to talk to you about how we've now advanced in the field to be able to determine peak endometrial receptivity. So this brings to mind the immediate questions of what is the endometrium and why test for endometrial receptivity. Well, as we all know, the endometrium is the uterine lining that builds up during a woman's menstrual cycle and is shed during menses if pregnancy does not occur. It is also the portion of the uterus that accepts an embryo for implantation and helps facilitate successful implantation. And the window of implantation is the period of time during this cycle where the uterine lining and the endometrium is most optimal to receive the embryo and facilitate successful implantation. Standardly, the window of implantation opens, if you will, around 5 days after initiation of progesterone during an HRT cycle, or 7 days after the LH surge during a natural cycle. However, all women are not alike, and all endometrium are not alike. And in some women, the window of implantation can be either delayed, meaning it happens after the dates that I just mentioned, or advanced, meaning that that window has opened and shut potentially before the embryo reaches the lining. And in fact, a recent study showed that in patients with recurrent implantation failure, or RIF, up to 43.5% of those women may experience a displaced window of implantation. And this, of course, could pose some serious challenges for successful implantation. So, the goal of molecular receptivity testing is to identify signatures associated with a receptive endometrium and to be able to identify before embryo transfer whether or not the woman would be predicted to have a receptive endometrium to facilitate implantation. And so what might cause a displaced window of implantation? Well, as we just discussed, there are some certain patient groups that may be more likely to have a displaced window of implantation. And one of these are patients diagnosed with repeated implantation failure. However, patients also with elevated body mass index, or BMI, or patients with adenomyosis might be more likely to experience displaced windows as well. Interestingly, for once a woman's window of implantation has been determined, it does remain. Some research has shown that it does tend to remain stable, potentially up over the course of 3 years and certainly over multiple IVF cycles. And this is felt to be true unless there are significant physiologic changes, such as an increase in BMI. So, to talk a little bit more about the ER-Peak endometrial receptivity test, its goals and its development, the goals of endometrial receptivity testing in the omics phase that we are currently in is to identify molecular signatures which are correspond to levels of expression, either up-regulated expression or down-regulated expression for specific genes that have been determined to be tightly controlled by hormones and correspond with either a receptive endometrium or one that is not quite yet receptive or has already passed that stage. And we in our laboratory looked through many studies from the primary basic science literature where genes that are hormone controlled and do correspond to a receptive endometrium, and identified about 40 such genes that seem to be the drivers of the expression profile that you would expect to see from either a receptive, pre-receptive, or post-receptive endometrium. And those form the basis of our panel and the molecular signatures that we are looking to identify with our ERPeak testing. We perform the testing via a validated RT-QCR platform. This platform is excellent and truly the gold standard for looking very specifically at gene regulation levels. And we've been utilizing this test with really good results to date. And now that we know a little bit about how ERPeak testing is performed, why might ERPeak be considered, particularly for repeat implantation failure patients? Well, as we've stated, this patient population is more likely to experience a displaced window of implantation, and thus having the benefit of ERPeak test results can help guide clinicians as to how to schedule that patient's embryo transfer for best results. And the format that our ERPeak results come back with is a simple 4-result category that can easily guide clinicians in that regard. As we'll talk in more detail about in just a couple of minutes, we have observed and studies have observed improvements in important clinical outcomes when using ERPeak in RIF patients. And as we touched upon briefly previously, our test has been found to have 5 times lower no result rates compared to other test providers, which is important to minimize the number of repeat biopsies that patients might have to go through in order to achieve a personalized or precision embryo transfer. So on to some of the outcomes that we've seen to date with the ERPeak test. And for here, I want to focus our attention on a recent study by Ohara et al., which was just released earlier this year. This study was a independent retrospective analysis that was conducted between April of 2019 and June of 2020 with 2 participating centers. And essentially what this study ended up doing was to compare 244 patients with repeat implantation failure who had received ERPeak testing and guided transfer versus 306 RF patients that had standardly timed embryo transfer. And importantly, I should note here that the definition of repeat implantation failure that was used in this study was patients that had 3 or more IVF cycles that failed to achieve clinical pregnancy after transfer of 1 or more or 2 high quality embryos. This study was uh, rigorously statistically controlled to ensure equal clinical characteristics between groups, and also was broken down further to look at outcomes between age groups of patients age 38 years of older or older versus patients under 38 years of age. And importantly, this was done because no pre-implantation genetic testing for aneuploidies or PGTA was performed in this for patients enrolled in this or involved in this retrospective analysis. To go a little bit further into the study design here, initially there were 1,000 patients that had received treatment that were retrospectively included in this study. 480 were in the arm that had received ERPeak testing, and 520 had not received that testing. As we had stated previously, of the patients that received ERPeak testing, about 55% were found to be of a receptive status, 43.5% were found to be non-receptive. In both sides of the study, there were a number of patients that were excluded from the ultimate analysis of clinical outcomes, and primarily on both sides of the study here, the exclusions were due to either the failure to create embryos of sufficient quality, or not having undergone a transfer of embryos for a number of reasons. And so we end up then with our group of 244 patients that had received ERPeak testing and personalized embryo transfer versus the 306 patients that had received standard transfer. To discuss what the findings were of the Ohara study and the clinical outcomes that were observed. The primary outcomes for this study were both clinical outcome and live birth rate. And for these categories, patients that received ERPeak testing that allowed personalized embryo transfer did better than the standard transfer cohort. Again, these were repeat implantation failure patients. And patients that received the ERPeak testing achieved a clinical pregnancy rate of about 38% as compared to a rate of 19% for the standard embryo transfer patients. On live birth rates, 30% of patients that underwent the ERPeak testing achieved live birth, compared to 9.5% of patients that did not receive that testing and the personalized embryo transfer. A secondary outcome that was looked at was miscarriage rates. And here, patients that, again, received the ERPeak testing and the personalized embryo transfer had a lower miscarriage rate of about 21% versus those who did not. And again, it's important to note here that the embryos that were transferred in this study were not subjected to PGTA analysis before transfer. And so to summarize the results of this study, with ERPeak testing and personalized embryo transfer, there were observations of significantly higher clinical pregnancy rates and live birth rates, as well as lower spontaneous abortion rates. For some additional secondary findings, as I mentioned, the authors further controlled for patient age, grouping patients into an advanced maternal age of 38 years of age or older, or less than 38 years old group. and then compared those for each of the against themselves for each of the arms as well. And it was still found that even when controlling for age, there were improvements in live birth rates when patients did receive the ERPeak testing versus if they did not. And there were quite striking additional improvements in the clinical pregnancy rates and the reduction of spontaneous miscarriage, particularly in those patients of advanced maternal age who were also RIF patients. Interestingly, there were 8 patients in this study that had received ERPeak testing that elected to have follow-up endometrial receptivity testing at a later date, typically just over a year after the initial testing had been performed. And for these 8 patients, 100% received the same result on the second ERPeak testing as they did on the initial ERPeak testing. So I hope this brings you up to date with where we stand with endometrial receptivity testing and the ERPeak test that we have developed within our laboratory. At this time, I'm happy to hand over to Tony Gordon to tell you about some exciting additional innovation that we have that we've recently concluded and released to you on other important aspects of the endometrium.
Thanks, Kristine, for that great walkthrough of endometrial receptivity testing. I'm going to move on to the latest part of our endometrial testing, and that's a microbiome test. And this is the other part that, if you pardon the pun, completes our ER-Complete test. So, the endometrial microbiome. I think this is a really interesting subject and one that we're delighted to be able to test. But we'll start off this by having a little bit of walk through the terminology in case it's new to you. So the microbiota, and this is the microorganisms of a particular site. It's the type of microorganisms. The thing that we use more often is, I guess, when we're talking about this, is the microbiome, and that's not just the organisms, but how they interact with their environment, because the organisms are not there just not doing anything. They interact with their environment. They have a biological niche, and they generate host responses, and that's what we're interested in looking at with this particular test. So for a long time, we thought that the uterine environment was sterile and that there was no microbiome there. But we've been able to use new molecular tests to look at the environment much more closely than we have been able to do before. And some of these papers are some of the basic papers that show the first reports of findings of microbes in the endometrial microbiome. And as you can see here, they describe this technology called 16S ribosomal subunit sequencing. And you can see that there's then consideration whether they've impacted success or failure. And over here on the right, this is from one of those papers, a little schematic, which shows, it's a bit hard to see because it's very small, but it shows that you can have a different composition of the different microorganisms that are there. And one of the ones that really was one of the first ones that we were concentrating on as an interesting one was lactobacillus. So lactobacillus is an interesting one because the amount of lactobacillus seems to have an impact on the environment and hence an impact on implantation in IVF. And you can see here from these two papers from Moreno et al. And Al Shah that high lactobacillus has an increased implantation rate, so over 90%, and a lower lactobacillus has a lower implantation rate. And it's interesting how that might occur. Uh, the lactobacillus, when it's high, seems to produce an environment that takes nutrients and produces lactic acid, deters other microorganisms, and makes the uterine cavity more um, appropriate for implantation. So it's an interesting test. So it's not just presence; it's percentage presence of lactobacillus. So we call this lactobacillus dominance, and this is one of the things, the first part of the ER biome. Um, just moving on to the next part. So detectable microbiome. So this is pretty interesting as well, because it's not just the lactobacillus. We're also looking for other microorganisms, pathogenic microorganisms that might create dysbiosis. And this paper from Moreno et al., which was published just last year, showed that about a third, just over 35% of the samples that they looked at had no detectable microorganisms. In our test, you can see here, internal data, we've looked at 242 samples here and have almost exactly the same proportion, 36.8, just 1% difference of samples where we couldn't detect a microbiome. And actually, that's an interesting one, because how do you confirm a negative? So if there's no microorganisms there, how do you know the test hasn't failed? And we've had to develop a test very carefully that has internal controls that can say, yes, the test was success, it passed its quality criteria, and it was successful, but there was no microorganisms present. So we think this is a really robust test, and actually having no microorganisms present seems to be quite favorable in terms, not as favorable as high levels of over 90% of lactobacillus, but still quite favorable in outcomes compared to having the pathogenic microorganisms present. So I'll just go to talk a little bit about our test development. The test that Kristine was talking about was a transcriptomics test. So that's looking at microRNAs, mRNAs, and looking for expression and how that then relates to particular proteins. This is a DNA test where we're looking for the presence of microorganisms. So how do we do this? So we spend a lot of time developing this test. First of all, we need an endometrial biopsy sample, of course. There is a slight question, can you do this on the same sample? So what we do is we split the sample, and we use a very small amount of the sample. We only need a tiny amount for the microbiome. And the first thing we do is extract the DNA, and then we amplify what's known as the 16S variable region. So 16S is a ribosomal subunit DNA sequence. It's best to think of it like a barcode for microorganisms. So it's highly variable, and depending on the sequence there, you can identify which microorganism is present and even subtypes of microorganisms. So we do the amplification, and then we do a DNA quantification to make sure we have DNA to test, or that our internal controls have worked. It's a really interesting area as well, because you have to be very careful not to have any contamination from the lab, any environmental contamination. So we spent a lot of time ensuring that we had as robust a test as this point. We then do a DNA sequencing using next generation sequencing, paired end. So we sequence from both ends, 600 base pairs, which is a long DNA sequence. You have a slightly different requirement for the ERPeak test, the transcriptomic test, to this, the microbiome test. We're looking for sensitivity, and we're looking for unknown microorganisms that we're trying to identify. So that's why we use DNA sequencing here. We then use a database, Silver, for these ribosomal RNA genes. Remember, it's a DNA test. And then we look to see which amplicon variants are there, and then we can identify here the different microorganisms that are present. And actually, it's a bit small, but the graphic shows the first 1, 2, 4, 5 are lactobacillus dominant. So the dark blue and light blue are lactobacillus, and then over here on the far right is a dysbiosis. So no lactobacillus, but a number of pathogenic microorganisms. This test has been validated very extensively, and looking at the accuracy and precision, you can see it's a really highly performing test. 99% accuracy in our technical verification and validation, sensitivity and specificity 95% and 100%, which is, I can tell you, really high performance for a test of this order. And then positive predictive value and negative predictive value of over 99%. Also looking at intra-run coefficient of variance. So if you run samples numerous times, just 1.2%, and then inter-run coefficient of variance, even less, 0.5%. So we've then looked at reproducibility across different sequencing instruments, the technical personnel, laboratories, run it in different laboratories, really to ensure that we have a very sort of robust design and protocols. So just to talk very little before we move on to talk about the complete test, about exactly how this could be used in practice. So it's a personalized fertility assessment to guide treatment by evaluating the endometrial environment. Initially, abundance of lactobacillus is really where the test has its main uses, and you can see here it determines the relative abundance of lactobacillus. So that goes back to that over 90% is favorable, under 90% non-favorable, or it identifies the presence of specific reproductive tract pathogens, bacteria. There's 18 pathogens that we look at, and it's all in the same test, so we can look for the lactobacillus, or we can look for pathogens all at the same time using this 16S ribosomal test and the first 3 variable regions. These are the 18 pathogens that we look at, and your report will give you a number of different outcomes. It will tell you whether there's microorganisms present or absent. It will tell you whether there's lactobacillus, a percentage of lactobacillus, or it will tell you whether these reproductive tract pathogens are present. And then it's up to the patients to talk to the clinicians to understand the next steps that they might want to undertake. So it's a really interesting test to add to offer in one testing both the endometrial receptivity and the microbiome. And with that, I'd like to pass back to Jenna, who's going to talk a little bit about exactly how the complete test works. Jenna.
Thank you, Tony. I'll try and finish up quickly here so we have plenty of time for questions, but I'm going to finish us off talking a little bit about the ER-Complete test and what is all entailed within. So the ER-Complete test, as the name implies, it encompasses both of the different tests that Kristine and Tony talked about. So it includes the ERPeak test, looking for that ideal window of implantation, and it also incorporates ERBiome, which includes looking at the relative abundance of lactobacillus as well as the presence or absence of reproductive tract pathogens. That's that 3-in-1. And the process of ER-Complete is relatively straightforward. It starts by the clinician performing an endometrial biopsy and preparing it for shipment. We request about 10 millimeters of endometrial tissue, and that's regardless of the test you're doing. If you're just doing ERPeak, if you're just doing ER-Biome, or if you're doing the whole complete both together, we still need just that same amount, about 10 millimeters of tissue. So that tissue sample is sent to the genomics lab, to our lab, where we will extract RNA for the ERPeak test and DNA for the ER-Biome element. We typically expect to issue a report within 14 days, and again, that's regardless of the test being ordered. And the report will let a clinician know whether that endometrial sample is considered receptive or not, pre-receptive, post-receptive, etc. If there is a pre or post-receptive test, there will be a recommended time for transfer, and it will also explain that relative dominance of lactobacillus and whether any specific pathogens were identified. So it's the process from the clinician's point of view. It's pretty much the same as the ERPeak test, if anyone is familiar with that and has ordered that before. So here is again just an overview of that ER-Complete and the 3 different elements, as well as some of the data. You can see the sort of takeaway from that Ohara study that Dr. McWilliams went over. Pregnancy rates were doubled in that study, birth rates were tripled, and miscarriage rates were more than halved. We also, with the ER-Complete test, we see very low no result rates. So we have evidence that the ERPeak test has a 5 times lower no result rate compared to other test providers. And the no result rate for ER-Biome is less than 1%. So low rates of getting an uninformative result. So you may be familiar with some of this complete language that you hear a lot when speaking with Cooper representatives. What is this complete? When we call a test complete, it means that it's our most comprehensive offering. So we have both PGT complete and ER complete. PGT complete includes our standard aneuploidy screening via PGT AI, artificial intelligence analysis involved there. It includes parental QC, which confirms that both intended gamete sources are represented in the embryos in question. There's the genetic PN check, which is really great for confirming that a 2PN embryo is a 2PN, a 3PN is a 3PN, a 1PN is a 1PN, etc. And the parental origin of aneuploidy element is included in complete as well. That's identifying if an aneuploidy arose in the egg or the sperm. And then, as we discussed, ER-Complete has those 3 elements, looking at the window of implantation, the abundance of lactobacillus, and the presence or absence of reproductive tract pathogens. Those are the complete tests. So with that, let's get into what I think is the fun part, which is the Q&A, putting our panelists here on the spot. So I'll invite both Tony and Kristine to unmute. Are you guys ready for some questions?
Hopefully so.
Awesome. All right. So as we've seen questions coming in, they kind of fall into 2 categories. There are some more procedural, logistical questions, and then some more scientific questions. So let's start with some of the logistical questions first. And maybe Kristine can handle a lot of these, but of course, Tony, if you have any particular opinions to share, please do. So the first question, and I sort of answered this one already, do you use the same sample to perform ERPeak and ER-Biome?
Well, Jenna, as you stated, I think you answered that beautifully, and Tony, I think you went into just in great detail and how we use the sample, but the answer is yes, we can use that same sample, about 10 millimeters of tissue, which we can separate as needed in our lab and send down the appropriate analytical pathway, whether it be an RNA-based analysis for receptivity testing or a DNA based analysis for biome testing. We are able to fully utilize that single sample.
Yeah, and I'll add to that. I know that in our lab we sometimes receive very large endometrial biopsies, and regardless of what tests are being done, just that 10 millimeters is enough. It doesn't need to be more than that. We don't really need a lot of sample.
Yeah, absolutely, Jen. And I'll just follow up on that as well. That sometimes those the very large samples can do more harm than good, particularly if in the efforts to achieve that sample, a lot of blood is introduced into the sample itself. That can unfortunately sometimes, though rarely, interfere with our ability to perform the testing.
That's true. That's true. So we want as clean a sample as possible if we can. Um, okay, so can these tests be done on different kinds of cycles, like IUI cycles, or non-IVF sort of cycles, or natural cycles? What kind of cycles are required?
Yeah. And I think there's a bit of a difference, important distinction if on what is ordered. If it is ER-Biome that is ordered, there's certainly much more flexibility in terms of the type of cycle, rather, if it's IUI versus IVF. But importantly, if the endometrial receptivity testing is going to be ordered either individually or part of complete, though it can be either an HRT cycle or a natural cycle, needs to be performed on a mock cycle that simulates what the eventual embryo transfer cycle plan will be. Because again, the goal of that test is to inform of whether or not in that mock cycle that mimics the planned embryo transfer cycle, whether the endometrium was in a receptive state. If so, then these same plans can be carried forward to the embryo transfer cycle. But if there was a displaced window of implantation, the ERPeak test will inform whether the embryo transfer cycle should be moved forward a day or perhaps moved back a day relative to what was originally planned.
Okay, so just to clarify for myself, for ERPeak, this does need to be a mock cycle where you're biopsying on P+5, or in a natural cycle LH+7, and therefore an ER-Complete also needs to be in a mock cycle because ERPeak is part of ER-Complete. But if it's just biome alone, does not need to be a mock cycle, correct?
That is correct.
Okay, good. And again, I'm seeing some questions about turnaround time. Again, for all tests, 14 days is the expected turnaround time. Okay, here's a good one. If someone has already sent a sample for ERPeak testing in the past, can they retroactively order ER-Complete or get that biome information from that same sample that was already biopsied?
And unfortunately, the answer to that is no. And I think that we've gone into some nice detail as to why that is, because in order to perform both tests at the time of receiving the sample rather, we need to be able to treat that sample differently, send part of it down one pathway, part of it down another pathway. We're not able to go back either in time or molecularly and get us back to that fresh sample.
If I'm not mistaken, we don't retain the original sample indefinitely. It is discarded after a relatively short period of time. All right. So if during ER biome or ER complete, if we do detect a reproductive tract pathogen, do we provide any recommendations for treatment in terms of things like antibiotics or probiotics?
Yeah, that's a good question. Um, I think it's important to note that our ER complete, ER, which includes ER biome and ER endometrial receptivity testing, these are screening level tests. And we recognize that our partner clinicians, physicians already have their own protocols in place for diagnosing and treating endometritis or supplementing, with lactobacillus, and we would really defer to their existing protocols for treatment.
Yeah, there are a few questions here about when you do have dysbiosis identified. Maybe Tony can answer this one actually. Are there any guidelines on the use of antibiotics before the biome test? Like, let's say someone has previously had an endometritis diagnosis and has had an antibiotic treatment for that. Like, when can they perform the ER biome test after treatment?
Yeah, it is a really good question, and it's one that I think is not just before the ER biome, but also if you've had an ER biome and you have antibiotic treatment directed by your clinician, whether you come back for another test in the future. Unfortunately, there's no real clear answer to that as things stand at the moment. We would recommend, you talk to your clinicians, see what they suggest. We don't have a specific recommendation for the period of time to wait for antibiotics.
Sure. And then kind of a similar question, and either of you can answer this. Uh, After testing and when there are pathogens detected, is it recommended to repeat the testing after treatment to see if the dysbiosis has been resolved?
Yeah, and I think there, Jen, I think what Tony stated is certainly still applicable in terms of ongoing study to understand, whether repeat testing would be either necessary or even beneficial. And I think that probably most clinicians already have their own practice in place about the steps that they take after treatment of endometritis. And I would say that even if incorporating ER biome testing into their protocols, that their existing protocols for managing patients after treatment would likely still apply.
Thank you for that. Back to Tony for a second. You spoke about this a little bit during your presentation, but let's just reiterate because it can be a little bit confusing. What is a no microbiome result? Is that a no result? Explain.
Yeah, it's a really good one because it's quite subtle but really important. So the difference between a no result and no microbiome found is is clearly distinct. If no microbiome is found, the test has still worked. It has still given you a result. It's not a test failure. If we have a test failure, that would be a no result on the report. And test failures we expect to be far less than 1%. The way that we distinguish that the test has worked is that we have controls within the procedure that have to be successful to show that the whole procedure has worked. And they show that things like the DNA amplification, the sequencing, the sequence analysis has all worked, but they've found no microbiome. So it's a subtle distinction, but the test has worked, and actually, it's probably good news for the patient that we found no dysbiosis or no lactobacillus, because it might not be as good as finding, over 90% lactobacillus, but it's still a fairly good result for the patient.
Interesting. I'm wondering, I don't know if this is a thing. Is it worth it to try and introduce lactobacillus? Is that something that can be done? Do you know?
Certainly, I mean, people do do that. I think, it's not something that we can advise on, and I think
Sure, sure.
that there's still lots of research ongoing. I think the research is pointing to benefit, but I think it's up to the patient and their clinicians to discuss it.
Makes sense. Um, Back to Kristine again, a question about the Ohara study and
Mhm.
how it looked at patients who had experienced recurrent implantation failure specifically. What do we know about the utility of endometrial receptivity testing in other groups of patients?
Mhm. Yeah, that's a really good question. To date, for our ER-Peak test, and that is again our test that looks at endometrial receptivity testing, which we developed um, on our own, with literature reviews, really targeting those around 40 genes that we really think are driving the transcriptomics, that molecular signature of a receptive endometrium. The Ohara study really is the strongest clinical study we've seen to date, and it is focused on recurrent implantation failure patients. We're really excited and hopeful to see multiple, additional studies come out in the near future that might look at other patient populations. But to date, the results that we discussed with you today are pertinent to the recurrent implantation failure
Okay,
population.
interesting. And kind of going along with that, can either of you speak a little bit to the evidence of stability of the window of implantation over time? Some may question, is it worth it to do it in a cycle? Is that truly predictive of the implantation window in a subsequent cycle?
Mhm. Yeah, absolutely. Yeah, and as I mentioned briefly, there are independent studies on this that have looked even with other endometrial receptivity tests and found stability between cycles for patients after endometrial receptivity testing. It was nice to see that corroborated, although on small scale, with the Ohara study that looked at 8 patients that did have the repeat testing, over a year later with ERPeak, and did show 100% concordance after a year with the ERPeak testing. So certainly there does seem to be evidence that is building up that the woman's endometrial receptivity status is somewhat stable, certainly over time, and also over fertility treatment.
I,
Okay.
I think you mentioned it actually in your presentation as well, Christine, that there are things that can impact endometrial receptivity like BMI. So
Mhm.
clearly that depends on other factors staying the same over that time period. So say the BMI changes dramatically, that might change the window of implantation. But yes,
Of
it's
course.
it's yeah it's something that I think we need more data on, but it looks good so far.
I agree. I agree. Yeah, and I think it's important to note that the studies like the Ohara one were fairly controlled in terms of even progesterone administration, and the protocol, and so certainly any changes to the HRT cycle could also impact the window of implantation.
Of course. So we're just about at time, but I really want to ask this one last question before we wrap it up. And this one is mostly for Tony, but again, Kristine, if you have anything to add, please do. So regarding the ER-Biome test, why are we not doing like a microbial culture? What's the difference between culturing and what we're doing, and why did we choose that?
Yeah, it's a great question, and it's an interesting area because this is an area where uh, genomics has really revolutionized microbiology because it's really hard to culture a lot of these microorganisms that you find in the uterine cavity. You know, anaerobic organisms and things like that are really hard to culture in traditional methods, and just the environment they are in might not be suitable to traditional culturing methods. So you don't get the full picture as you do, and you don't get the accuracy as you do from next generation sequencing. So next generation sequencing can give you all these different pathogens, their proportion in the biopsy. They don't tell you they don't tell you whether the pathogens are alive. they tell you whether the presence of DNA is there, but it's still really useful information. So that's why we move on to doing next generation sequencing for this rather than traditional microbiology methods. But having said that, they are the gold standard if you really want to confirm this, and that's something you'd need to discuss with your clinician for your patient.
Very interesting. Well, thank you so much for sticking with us to the end there. That was such an interesting discussion, really at the forefront of genomics, and there are just so many new and interesting things coming out. And I'm sure that Tony and Kristine and I will be here talking about some new interesting development in the coming months and years. So thank you so much for joining us for this webinar today. Thank you for your questions, all the interesting topics that were raised. And again, please join us in our future webinars and be well.